Okay, hello to all of you. I am now going to discuss the protein isolation lab number two. So we have basically in the first lab we have isolated proteins with the help of the technique called ammonium sulfate precipitation. And what we have till now, we have saved filtrate, we have saved S1, we have saved S2, and we have also had two pellets, P1 and P2, and those two pellets we started dialysis with them. And next week, when you come back, your P1 and P2 will be ready, they will be dialyzed already. And what we have to do in the second lab is we are going to actually we're going to purify the protein. So our goal of this lab is to isolate rubisco. So we are working on a special protein called rubisco, which is involved in photosynthesis, and we have used the spinach leaves. So with the ammonium sulfate precipitation, we have precipitated the proteins, and it's a mixture of proteins, but we are interested in our specific protein. So what we have to do now, we have to pass the mixture of proteins which is there in our pellet, and those two pellets we have dialyzed because we wanted to remove the salts from them. So those pellets which has a mixture of proteins, we will be passing them through a column so that we can get our desired protein, which is rubisco, and we'll all these according to flow columns. And so basically these columns, they will have a resin in them, and that resin consists of tiny beads of cellulose that are covalently bound to positively charged side chains. Now the column which is made is basically having the beads of cellulose, and they are attached to some side chains, and they are positively charged. So what is going to happen is when we are going to pass the proteins over this column, all the negatively charged proteins, they will be attaching to the column, and everything else is going to pass through the column. So we are going to then eventually elute the proteins which are attached to the column. So remember that column is positively charged, and the negatively charged proteins will be attaching to the column. They will be attaching to the column, and then all the other proteins, they're going to pass through the column in which we are not interested. So our desired protein is actually bound or is actually attached to the column, and then eventually by using some buffers, there's a low salt buffer, medium salt buffer, and high salt buffer, we are going to elute. So the word is elute, and we are going to do illusion. So by that we are going to basically separate the protein which has been attached to the column, and that will be our desired protein. And then with that protein we are going to run the gel and along with other samples. So I will tell you. So let's go over the technique. So basically the columns will be given to you. It will have some parts. So basically the column has the plastic column, filter, stopcock, and lid. You will be actually getting the columns ready made. You don't need to prepare the column, it will be ready made for you, and I will show you in the lab what this column is and how to fix the column in the stand and all that. These things I will show you in the lab. So basically the very first thing what you're going to do is you are going to equilibrate the column by applying 30 ml of buffer A. So basically you will be given a buffer A, and you have to pass this buffer over the column, and this is called equilibration. So you're passing this buffer over the column, and your column will be getting equilibrated. And make sure that in this whole process, a very important precaution is your column should never get dry. So this is a very, very important precaution. Okay, once the equilibration is done, what are you going to do? And also, so the equilibration will take at least 10-15 minutes, because you have to pass around 30 ml of buffer A through the column. At that time, what you have to do, or you can divide the work in your group. So you have to basically spin the P1 and P2. So the P1 and P2 I will be giving you, which is in your dialysis bags. You are going to take your P1 and P2 in separate tubes. You are going to spin them, because if anything is there, we would like that thing to get settled. Any solid precipitates or something, if it is there in your P1 and P2, we want them to settle down. So for that, what are you going to do? You are going to do the spinning at a very low speed, and which is 1000 RPM for 3 minutes. Okay. So, and then what you're going to do after you have done the spinning, you will collect the supernatant, and very, very important thing here is you need to save 1 ml of each P1 and P2 in the microcentrifuge tubes or Ependorf tubes, and we will be saving them in the fridge, because next week when we run the gel, we definitely need P1 and P2 before passing through the column. So you can label them as BP, like a P1 and P2. BP means before purification. So we are saving 1 ml of P1 and P2 before purification. Very important sample, because that way we can see on the gel if our purification has worked or not. So after purification, you should be able to see less proteins, and before purification, there should be more proteins. So these two tasks will go side by side. You will start equilibrating your column, and you will start spinning your P1 and P2. Now what you have to do next is dilute the, so whatever the P1 you have, what you're going to do, you are going to dilute it. So P1 is quite concentrated. Here it's written 100-fold, but I would like all of you to make a correction here. This is in our experience in the past. We have seen if we dilute it 100-fold, we don't actually get good amount of protein in the when we collect the purified protein. So make a change here. Do the dilution 25 times, so it will work good. So and you are going to pass around 5 ml of each sample over the column. So after it is equilibrated, you're going to pass 5 ml of diluted P1 and undiluted P2. Okay, so once it's done, allow the samples to flow into the column. Then wash the columns by passing 10 ml of buffer A through the column. Okay, so after that you have to do the washing. So wash columns by passing 10 ml of buffer A through the column. And then next what we have to do, so we have equilibrated the column, we have our samples ready, and we have also passed the samples over the column, and then we have also washed the columns. After that, now it's a time to do the elution. We are going to elute the proteins. So now when we have passed our sample over the column is positively charged, and the proteins, our Rubisco is negatively charged, and it is going to go and bind to the column, and all the other proteins, they're going to pass through the column, and we're not interested in those proteins. So when you wash, everything is going to go out which is not desired, and our desired protein is now sticking to the column, and now it is our time to elute it. Eluted means we are going to remove our desired protein from the column, and that we do by using 3 different kinds of buffers. We use a low salt buffer, we use a medium salt buffer, and we use a high salt buffer. So you will be eluting the proteins using 3 buffers, and you will be collecting the fractions. And then side by side, you also need to measure the OD at 280 nanometers, and you will be having, like, for each elution you will have around 4-5 fractions. You need to check the OD in the spectronic, and you need to see which sample has good amount of OD. Usually if the OD is more than 0.1, we will keep that sample. If the OD is less than 0.1, you can just throw that, you can just toss it. We don't need that because it has a very, very low amount of protein. So another important thing I would like to tell you here is why we are using OD at 280 nanometer. It's in the UV range. Some proteins, the proteins like Rubisco, basically they have amino acids which are aromatic, like tryptophan, phenylalanine. These are all amino acids which are aromatic ring structures, and you will start collecting these fractions. You will start collecting these fractions, and you will measure the absorbance at 280 nanometers. And remember that what kind of buffer you are using to do the elution. You will be using the same kind of buffer for blanking the speck. When you are doing the elution with low salt buffer, the blank will be low salt buffer. When you are doing the elution with medium salt buffer, the blank will be medium salt. When you're doing the elution with high salt, the blank will be high salt. So you will do the same thing with the medium salt buffer as well as the high salt buffer. And for each kind of a buffer, when you're passing through the column, you are going to collect the fractions, and you are going to take the OD in this spec, and you are going to save only those fractions which have at least OD more than 0.1. So label your tubes, write it down which fraction it is, from which kind of buffer it is, and you will be saving all these fractions in the box, and we are going to keep that box in the fridge. Actually, we can use the same box which we have been using, and this way you will have collected all your fractions. So do not forget that you have to save P1 and P2 before purification, and then you have to save all the fractions which are coming from different buffer illusions, and you will be keeping the samples which are more than 0.1. So this is what we are going to do, and then you can basically clean the resin. This buffer will be given to you, and we will see if we need to clean it or not, or the lab people will take care of that. So this is basically we are going to do this week. And yeah, and the important thing to know for this week is the discussion point is you should know what is ion exchange chromatography. Okay, so we are using ion exchange chromatography this week, so you should know what the principle behind it. So that's all for the next week's lab, and I will see you guys in the lab.